Hi everyone, this is Shakil Ahmed, your biology educator, and I'm here with one more topic on our Cambridge biology syllabus. Here we go. And I am here with the latest paper. It's Summer 22, Variant 32 for the AS practical exam. I've selected question number 2 of this paper because this question seems to be a little different from other questions of microscopy that we usually get. It is different in the sense that we have a certain portion shaded in the figure and that we have to make the plant diagram of. So let's go directly to the question. Question number 2 says that K1 is a slide of a stained microtransverse section through a plant stem. Draw a large plant diagram of the region of the stem on K1 indicated by the shaded region. In figure 2.1, use a sharp pencil and use one ruled label line and label to identify the epidermis. Now, because we don't have the microscope available here and we cannot show you the image that is seen in the microscope, I'm just taking the usual, we have the TS of a dicot stem. So, in fact, they are telling us to make, because if this is the slide that we see in the microscope, we, uh, we usually focus it at power 10 when we have to make the plant diagram. So we will select this portion of the slide that we have to make. So we'll focus on this slide portion on the microscope and then start making the plant diagram of this portion. So remember one thing, that we have to use more than 75% 3 by 4 of space that's available to us. So we will start making the drawing like this one. If this is the portion that we have to make, we can clearly see that we see the epidermis over here. So we'll show the two lines showing the start and the end of the epidermis. Then we can show the vascular bundles being seen over there. And then in between the vascular bundles, there is a tissue growing like this one. And we have the peak also being shown over here. So this is the plan diagram of this slide. Remember, we don't have to write anything below this line where the marks are marked, and we have to take care of the lines, pencil line. They must be quite sharp. The lines must not be feathery. They must not be overlapping, and we must not actually. The lines must be very clear. So this is how the marks are distributed in the plan diagram. Then moving to the next part of this question is, observe one of the four large vascular bundles of the section on K1. Select a group of four adjacent xylem vessel elements. Each xylem vessel element must touch at least two other xylem vessel elements. Make a large diagram of this group of four xylem vessels and use one ruled label line and label to identify the wall of one of the xylem vessel elements. Now, what we see here is when they tell us that a cell must touch at least two other cells. First thing is, whenever we have to make the individual cells, we select the power 40 on the microscope. And when we have to show that each cell touches at least two other cells, that means the cells must be in the two lines, on the two rows. So if one of the cells is like this one and the other is like this one, the other two cells must be over here, so that the criteria they have set for the question is met. Each cell touches at least two other cells. Because these are the plant cells, we always make the double wall to show that it's a cell wall also surrounding them, or the cell membrane and the cell wall. The same criteria is allowed or applied over here as well, that we have to use more than 75% of the space that is available to us. Second thing is that the diameter of each plant cell or in individual cells should be minimum 40 to 45 mm. So they are showing us, asking us to make the cell wall. Okay, let's see. This is the cell wall of one of the xylem vessel elements. Now moving to the second another part of this question. Figure 2.2 shows a diagram of a stage micrometer scale that's being used to calibrate an eyepiece gratitude. The length of one region of this micrometer scale is 1 mm. So this means it's 1 mm from here to here, one from here to here, one from here to here, one from. So we have 1, 2, 3, 4, 5, 6. This is quite unusual to have 6 mm or 1 mm division on a micrometer stage micrometer, because usually what we have is each division on a micrometer scale is zero point 0.1 mm, and we have a total 0.25 mm available, means zero point 0.1, zero point 0.1, and the last one 0.25. Anyways, this is a different type of stage micrometer they are actually asking us to use. It has 6 mm divisions, so we have to use figure 2.2 calculate the actual length of one eyepiece graticule, show you a working. We can show this working in a different ways. Like I can see it here, 100 eyepiece graticule units is equal to 6 mm. So how much is one eyepiece graticule equal to? So we can say it will be 1 over 6 over 100, 0.06 mm. We can convert it into micrometers multiplied by 1000, which will be 60 micrometers. Or we can see it from here. If it's this is coinciding exactly eyepiece graticule, we can see it that 1 mm is equal to 16 eyepiece graticule units. So how much will be one eyepiece graticule unit equal to? It will be 1 over 16, the same thing. So we'll have 0.06 mm multiplied by 1000 means 60 micrometers. Figure 2.3 shows a photomicrograph of a transverse section through a different stem to K1. This was taken with the same microscope and lens is used to take figure 2.2. The eyepiece graticule has been placed across the diameter of the section. Use the calibration of the eyepiece graticule from B1 to calculate the actual diameter of the section in figure 2.3. So what was the calibration that we calculated in B1 was one eyepiece graticule is equal to 60 micrometers. Now, moving to this question, let's see what is the diameter of this cell. If we see, we start from 6 over here and we end at 80, means 80 minus 6, 74 eyepiece graticule units is the diameter of the cell. We have to find the actual. So because one eyepiece radical unit is equal to 60 micrometers, so we can say 74 into 60 means 6 fours are 24, 6 sevens are 42, plus 2 is 44. It will be 4,440 micrometers. That's all for this session. I'll be back with you with another session with some interesting and challenging paper. Thank you very much.